The leaves of plants are the primary site of photosynthesis. A cross-section of a typical dicot leaf shows that its surface is composed of epidermal cells. The epidermis may produce leaf hairs that protect the leaf by manufacturing irritating substances. The epidermis also regulates the leaf's heat and water balance. A waxy substance called the cuticle covers the epidermis of the leaf and protects the leaf from drying out. It also prevents the uncontrolled exchange of gases. The controlled exchange of gases occurs through openings in the epidermis called stomata. Each stoma is flanked by 2 guard cells. When the guard cells are turgid, the stoma is open. The stoma closes when the guard cells. The interior of the leaf is filled with mesophyll tissue. The layer of tightly packed mesophyll cells is called the palisade layer. These cells are specialized for light absorption. The palisade mesophyll cells are densely packed with chloroplasts and perform most of the leaf's photosynthesis. The layer of loosely packed mesophyll cells is called the spongy layer. Although photosynthesis also occurs in spongy mesophyll cells, this layer of cells is specialized for gas exchange. Air spaces throughout the spongy layer increase the area available for gas exchange. The air spaces connect with stomata, which control the flow of gases into and out of the leaf. Leaf veins pass through the mesophyll tissue. Xylem and phloem are the 2 types of vascular tissue found in leaf veins. Bundle sheath cells surround the vascular tissue. Xylem transports water and dissolved minerals from the roots to all parts of the plant. The elongated cells that make up the xylem are dead at maturity. The thick cell walls help prevent the cells from collapsing and also help support the plant. The water is pulled up through the xylem to replace the water lost through evaporation from the surface of spongy mesophyll cells. The phloem transports carbohydrates and other organic materials throughout the plant. Unlike the xylem, the phloem is made up of living cells. During photosynthesis, energy from sunlight is used to convert carbon dioxide and water into sugar and oxygen. This series of reactions occurs in the chloroplasts of mesophyll cells. Water is carried into the leaf by the xylem and diffuses into mesophyll cells. Carbon dioxide enters the air spaces in the leaf through open stomata and diffuses into mesophyll cells. Once inside mesophyll cells, the water and carbon dioxide enters chloroplasts. Sunlight also enters chloroplasts, and its energy is used to produce sugar and oxygen from carbon dioxide and water. The sugar moves from the mesophyll cells into the phloem and is transported away from the leaf. The oxygen diffuses out through open stomata. A closer look at one of the mesophyll cells shows that it contains several organelles, including a nucleus, mitochondria, and a large central vacuole. The central vacuole plays an important role in maintaining the internal water pressure of the leaf. The cells of the palisade mesophyll and spongy mesophyll also contain chloroplasts, the tiny organelles where photosynthesis takes place. The chloroplast is surrounded by a double membrane. The internal membrane is organized into flattened sacs called thylakoids. A stack of several thylakoids is called a granum. The fluid surrounding the stacks of thylakoids is called the stroma. Thylakoids contain photosynthetic pigments such as chlorophyll that capture the energy in sunlight. Once harvested, this energy is used to produce ATP and NADPH. Energy from ATP and NADPH is used by enzymes in the stroma to incorporate atmospheric carbon dioxide into organic molecules. More than 95% of the water gathered by a plant evaporates back into the atmosphere from leaves and stems, often only a few hours after being absorbed. The evaporation of water from the shoot of a plant is called transpiration. Water moves rapidly into the roots, is pulled upward through the root and the stem into the leaves to replace the water lost through transpiration. A capillary-sized pipeline called the xylem maintains the cohesion of the water column as it travels through the plant. Most absorption of water occurs just above the growing root tips where the root hairs are located. A cutaway view exposes the arrangement of cells in the root. The outer layer of cells is the epidermis. Extensions of epidermal cells form root hairs. Other tissues include the cortex, the endodermis, containing the Casparian strip, and the vascular tissue. There are two types of vascular tissue. Phloem transports organic nutrients, usually from leaves to the roots. Xylem transports water and minerals from the roots to the leaves. Water can reach the xylem by either of two different pathways. Using the symplastic or intracellular pathway, water passes through the selectively permeable membrane by osmosis and travels into the cytoplasm of the epidermal cell. The water then passes through openings called plasmodesmata directly into the cytoplasm of the adjacent cortex cell. In this manner, water moves from the cytoplasm of one cell to the next without crossing cell walls or membranes and enters the endodermis. From the endodermis, water moves into the xylem. Alternatively, water can take the apoplastic pathway, squeezing through the spaces between cells or traveling through the cell walls of adjacent cells. Reaching the endodermis, the water encounters a band of waxy material in the cell wall of the endodermis cell. This band, called the Casparian strip, is impermeable to water and dissolved substances. Therefore, the Casparian strip directs water through the selectively permeable membrane and into the endodermis cell. Thus, the endodermis regulates the movement of water and dissolved material into and out of the vascular tissue. The xylem is made up of two types of water-transporting cells, the long, thin tracheids and the shorter, wider vessel elements. Both types are hollow and non-living. Water moves from one tracheid to another through pits or depressions in the cell walls. The end walls separating adjacent vessel elements, on the other hand, are wholly or partially dissolved. Therefore, water moves through a continuous pipeline formed by vessel elements running up the stem of the plant and ultimately into the leaves. Since water is a polar molecule, the individual water molecules adhere to each other, forming a continuous column of water from root to leaf. The column of water is pulled upward into the leaves by water loss due to transpiration. Inside the leaf, water from the xylem passes into the spongy mesophyll cells. Less water is present in the air than in the cells of plants. Therefore, water evaporates from the surfaces of the cells and enters the pockets of air scattered between them. Finally, water diffuses out of the leaves through open stomata. Sugars are manufactured in the leaves of plants by photosynthesis. This area of the plant is called the food source. Sugars produced at the food source are transported through the phloem to the part of the plant where they are used or stored. Using the potato plant as an example, the sugars are converted into starches and stored in the potato, which is a tuber on the root. This area is called the food sink. In the leaf, sugar synthesis takes place in the chloroplasts of mesophyll cells. The sugar molecules move through plasmodesmata from one mesophyll cell to another toward the phloem in the leaf vein. The sugars pass through the bundle sheath cells and into parenchyma cells in the phloem. The sugars enter the companion cells. Next, the sugar molecules are actively pumped into the sieve tube cells. The removal of sugars from the companion cells produces a concentration gradient. Therefore, sugars diffuse from the mesophyll cells, which are the food source, through phloem parenchyma cells and into companion cells. Companion cells continue loading sugars into the sieve tube. Water also moves along this concentration gradient and flows from the xylem into the sieve tube by osmosis. This influx of water increases the pressure in the sieve tube. The increase in pressure forces both water and sugars to flow along the sieve tube from cell to cell by way of sieve pores occurring in the common wall between the tube cells. Sugar is unloaded at the food sink, such as within a potato, where it is converted for storage or used for energy. Water also leaves the sieve tube since it too is used by the food sink and surrounding tissues. In addition, some water makes its way back into the xylem and is recirculated. Plants whose leaves drop seasonally are said to be deciduous. In northern temperate zones, deciduous plants shed their leaves and enter a period of dormancy to survive the low temperatures of winter. New leaves are produced in the spring when day length increases. They grow and mature through summer. This is the leaf maintenance phase marked by high levels of the plant hormone auxin. The leaf remains green, healthy, and fully functional. The chloroplast, mature leaves, contain several kinds of colorful pigment molecules that play a role in photosynthesis. Chlorophylls are green, while carotenoids are orange or yellow. Phylls are red. All of these pigments are broken down and resynthesized regularly throughout the growing season. However, the chlorophylls are usually present in much larger quantities than the other pigments. As a result, the orange, yellow, and red pigments are masked by the green ones. Therefore, the leaf appears green. Decreasing in early fall induces a change in the balance of hormones produced by the leaf. Particularly important is the drop in auxin and accompanying rise in estrogen that characterizes the shedding induction phase. A number of changes occur in the leaf at this time. An abscission zone begins to form of the leaf petiole. This zone consists of two distinct layers. The separation layer develops on the blade side of the abscission zone. This layer is composed of relatively short cells with poorly developed cell walls that make them structurally weak. The protective layer develops on the side of the abscission layer closest to the stem. The cells of the protective layer become impregnated with a fatty substance called suberin. In addition, the rate of chlorophyll synthesis slows and then stops. The orange, yellow, and red pigments in the chloroplast show through. Other brightly colored pigments, such as anthocyanins, are produced outside the chloroplast, intensifying the color. Finally, nutrients such as amino acids and sugars are translocated to the stem. The shedding phase begins when enzymes are synthesized that digest the structural elements of the cells in the separation layer. A gap forms between the two layers, and soon all that holds the leaf in place is a few strands of xylem. Stripped of reusable substances, the leaf contains little more than cell walls and remnants of cytoplasm. A gust of wind can easily break the connecting strands, and the leaf falls. All that remains on the stem is the leaf scar, formed by the protective layer. The suberin in the protective layer guards the stem from infection and excessive loss of water. A period of dormancy begins. Dividing cells can be found in the growing leaves, roots, and stems of plants. Several important features of a plant cell are the cell wall, cell membrane, cytoplasm, nucleus, nucleolus, and nuclear envelope. Most of the time in its life cycle, a cell is in interphase, the phase between successive mitotic divisions. During this time, the DNA is dispersed within the nucleus in the form of delicate thread-like strands called chromatin. The DNA replicates toward the end of interphase as the cell prepares to divide. Mitosis begins with prophase. During prophase, the chromatin strands condense until individual chromosomes are visible. Since the DNA replicated before mitosis began, each chromosome is composed of 2 sister chromatids. The chromatids are attached at a constriction point called the centromere. Protein complexes called kinetochores develop at the centromere. Simultaneously, the nucleolus and nuclear envelope disappear. Microtubules assemble to form the spindle apparatus. Some spindle fibers from opposite poles of the spindle attach to the kinetochore on either side of each chromosome. During metaphase, the kinetochore fibers align the chromosomes in a plane midway between the poles of the spindle apparatus. This plane is called the metaphase plate. Anaphase begins when the kinetochore fibers shorten, separating the sister chromatids and pulling them to opposite poles. By the end of anaphase, the 2 identical sets of single-stranded daughter chromosomes have moved to opposite poles. During telophase, the spindle apparatus disappears, and each set of daughter chromosomes is surrounded by a nuclear envelope. The chromosomes uncoil and again become the thread-like strands called chromatin. Cytokinesis begins. This process divides the cytoplasm and results in the formation of 2 separate daughter cells. A system of fibrils, called the phragmoplast, appears between the 2 nuclei. The fibrils of the phragmoplast, like fibers of the spindle apparatus, are composed of microtubules. Small vesicles appear at the midline of the cell and fuse, forming the new cell wall or cell plate. Like an expanding donut, the phragmoplast grows outward, and the cell plate grows as vesicles are added to the outer edge of the central vesicle. The cell plate continues to grow until it fuses with the plasma membrane of the parent cell, separating the 2 new daughter cells. The new cell walls formed in the central vesicle join the cell wall of the parent cell. The daughter cells lay down additional cell wall material. The cell wall of the parent cell loosens and tears as the daughter cells enlarge. One cell matures and elongates, while the other will divide again. The quantity of matter that makes up the Earth is fixed and finite. For life to continue, this matter must constantly be recycled within and between the Earth's ecosystems. Carbon and nitrogen are 2 of the most common elements found in living things. Therefore, the carbon and nitrogen cycles are essential for life. The carbon cycle mainly involves the exchange of carbon between the atmosphere and organisms. Through the process of photosynthesis, plants use the sun's energy to combine carbon dioxide from the atmosphere with water. Carbohydrates are produced that can either be used as fuel or to build other organic compounds. The energy contained in these organic compounds is harvested by both plants and animals through the process of respiration. During respiration, oxygen is consumed and carbon dioxide is released into the atmosphere. Carbon incorporated into the body of an organism may not be returned to the atmosphere until the organism dies and its body is decomposed by fungi and bacteria. These organisms use the organic compounds found in the dead body as fuel. Through respiration, they also release carbon dioxide into the atmosphere. Carbon is removed from the cycle for even longer periods of time when animal and plant remains become buried before they are completely decomposed. For example, millions of years ago, During the Carboniferous period, trillions of tons of plant and animal material were buried. Over time, the organic material was compressed and heated, resulting in coal, oil, and other fossil fuels. By digging into these reservoirs and burning this fossil fuel, we harness the energy contained in this organic material. We also release the carbon dioxide and water removed millions of years ago from the atmosphere. As we have seen, carbon dioxide is removed from the atmosphere through photosynthesis. The organic compounds produced may become incorporated into the bodies of plants and animals. Dead plant and animal decomposed or become buried in sediments. Carbon dioxide is again released into the atmosphere when the energy contained in these organic compounds is used. The nitrogen cycle is also essential for life, since nitrogen is a component of both proteins and nucleic acids. About 79% of the air is composed of nitrogen gas, but plants and animals cannot use nitrogen gas directly. Atmospheric nitrogen must first be combined with other elements. This conversion takes place in three ways: industrial fixation, atmospheric fixation, and biological fixation. Industrial fixation takes place when energy is used to produce fertilizers that contain ammonia, ammonium, and nitrate. Farmers, gardeners, and homeowners increase plant production by adding these fertilizers to soil. Atmospheric fixation takes place during combustion, volcanic activity, or thunderstorms. During thunderstorms, for example, extremely high temperatures associated with lightning cause a small amount of nitrogen to combine with oxygen and hydrogen to form nitric acid. Dilute nitric acid falls to the ground as rain or snow. In the soil, nitric acid is converted to nitrate. Although both industrial and atmospheric fixation increase the pool of available nitrogen, the bulk of nitrogen fixation takes place as a biological process. Biological fixation of nitrogen is carried out by nitrogen-fixing bacteria. Some of these bacteria have a mutualistic relationship with certain plants and are closely associated with the roots of these plants. The nitrogen-fixing bacteria produce ammonia. The ammonia is absorbed by the plants and used to synthesize nitrogenous molecules such as amino acids. Amino acids are then combined to make proteins. Other nitrogen-fixing bacteria are free-living in the soil. These bacteria also convert nitrogen gas to ammonia. The ammonia is released into the soil, where nitrite-forming bacteria combine ammonia with oxygen, forming nitrite. Still other bacteria, called nitrate-forming bacteria, convert nitrite to nitrate. Plants absorb the nitrate, convert it to ammonia, and use the ammonia to produce amino acids and other nitrogenous compounds. Animals obtain their nitrogen by ingesting plants or other animals. Nitrogenous food molecules, such as proteins, may be incorporated into the animal's tissues, converted to other molecules, or used as a source of energy. Many mammals convert the excess nitrogen from these uses to urea and excrete it. When animals and plants die or simply shed structures such as hair or leaves, a variety of decomposers break down the organic molecules. Among these decomposers are ammonifying bacteria. These microorganisms convert nitrogen-containing molecules into ammonia, thus making the nitrogen available once again to other organisms. Most of the nitrogen cycling through natural systems moves between soil, water, and organisms. A small portion of this cycling nitrogen is returned to the atmosphere by denitrifying bacteria, which use nitrate as a source of oxygen and release nitrogen gas. Nitrogen fixation serves to increase the amount of nitrogen available to plants. An ecosystem is a stable ecological unit composed of a living, biotic component of interacting organisms and a non-living, abiotic component that consists of physical factors of non-living parts of the environment. To be maintained, an ecosystem must have a steady input of energy. The source of energy for most ecosystems is the sun. The sun's energy is trapped and then transferred from one organism to another. The sequence of organisms through which energy and nutrients flow is a food chain. The food chain begins with plants, which are the producers in the food chain. During photosynthesis, plants use the sun's energy to produce sugars and other organic compounds from the atoms found in their environment. Thus, in our example, plants are the first class of organisms to capture energy entering the ecosystem and use it for growth and reproduction. In relation to this flow of energy, the plants are classified as trophic level 1. The energy in trophic level 1 can be transferred to other organisms when the plants are consumed. The animals that eat producers, such as plants, can be referred to as primary consumers. Primary consumers make up trophic level 2 in the food chain. Mice are the primary consumers in our example. Animals called predators get their energy by eating other animals. A predator, such as a snake, becomes a secondary consumer when it eats a mouse who was a primary consumer. Secondary consumers make up trophic level 3. The animals in the third trophic level are often food for other predators. In our example, hawks prey on the snakes and thus form trophic level 4. They are the top predator in this food chain since no other predator eats them. So in this example, the food chain consists of 4 trophic levels. As energy passes from one trophic level to the next, approximately 90% is lost. This flow and loss can be expressed as an ecological pyramid. Ecological pyramids may represent either the number of organisms, the biomass, or the amount of energy found in each trophic level. Commonly, these factors are directly related, and, as our example shows, each gets smaller and smaller as we process through the pyramid. Eventually, all the energy in living systems is lost as heat and cannot be recaptured. Therefore, energy does not recycle through a system, but flows through it. Ecological pyramids are also related to another important ecological principle, biological magnification. Because a single organism at a higher trophic level consumes many individuals from a lower trophic level, contaminants in the environment tend to become more concentrated or magnified as they make their way up through a food chain. For example, a persistent insecticide, such as DDT, may be present at very low concentrations when applied and may enter the tissues of plants without affecting them. When the plants are eaten by animals in the second trophic level, the insecticides are not broken down but accumulate in the animal's tissues. As the animal eats more plants, more and more insecticide builds up. The insecticides become even more concentrated in animals at the third trophic level. Again, the concentration of insecticides increases in the fourth trophic level. At these high concentrations, the insecticides may become toxic. As a result, the balance of the food chain may be seriously disturbed. The continents of today were originally part of a single supercontinent called Pangaea. The supercontinent broke up, and the pieces drifted into their present positions. This movement of continents is called continental drift. The history of continental drift is explained by the theory of plate tectonics. According to this theory, the Earth's crust is broken into several individual pieces called plates. These rigid plates move very slowly but continuously. The continents, as part of these plates, move as well. The movement of tectonic plates has been a major factor in producing the distribution of life forms throughout the history of our planet. For example, members of the group of flightless birds called ratites are native to southern continents and islands that are today widely separated. Ostriches are found in Africa, rheas in South America, and emus in Australia. These flightless birds diverged from all other living groups of birds at a time when the southern continents were connected. Ratites migrated freely between the southern continents until the continents drifted apart, isolating the ancestral population of ostriches, rheas, and emus. Monkeys with prehensile tails are found only in Central and South America, but not in Africa. This distribution implies that no overland migration routes to other continents were present after these groups evolved. We are seeing the Earth as it appeared toward the end of the Paleozoic era during the Permian period between approximately 280 and 250 million years ago. The northern part of Pangaea, called Laurasia, was composed of North America and Eurasia except for the Indian Peninsula. The southern part, called Gondwana, was composed of Africa, South America, Antarctica, Australia, and the India Peninsula. The fossilized remains of mammal-like reptiles called therapsids are known from all the continental regions. Striking similarities between the faunas of Russia and South Africa, for example, indicate that similar ecological and climatic conditions existed between Europe and Africa. Because these animals lived in the open plains and were not adapted for swimming, their distribution indicates that similar ecological and climatic conditions as well as land connections were present. Evidence linking the continents that form Gondwana is provided by the occurrence of the Permian seed fern called Glossopteris. This plant grew in South America, South Africa, Antarctica, Madagascar, India, and Australia during the Permian period. Although some of the continents began drifting apart at the start of the Mesozoic era during the Triassic period, extensive land connections were still present. During this period, between approximately 248 and 213 million years ago, a number of reptiles and amphibians had worldwide ranges. For example, labyrinthodon amphibians can be found in the Triassic deposits from Germany through the middle latitudes of the northern and southern hemispheres and into Antarctica. In addition, the fossils of a carnivorous wolf-like therapsid called Cynognathus have been found in parts of South America as well as Africa and Antarctica. The continents began to drift farther apart. During the Jurassic period, approximately 213 to 144 million years ago, the landmass that was to become southern India split off from the rest of Pangaea. The rift between Africa and the joined continents of Antarctica and Australia widened. Laurasia moved away from South America, and Africa began to move north. Despite the spread of Jurassic seas, the similarity of dinosaur faunas found in Europe, North America, and Africa at the end of this period indicates that environmental conditions were similar in these areas and intercontinental connections were present. The continents continued to drift. By the Cretaceous period, approximately 144 to 65 million years ago, Australia and Antarctica had separated, and India was nearly at the level of the equator. South America and Africa were widely separated. North America and Eurasia began to separate. Marsupial and placental mammals probably both evolved during the Cretaceous period. Marsupials appeared in South America and spread through Antarctica and into Australia. Placental mammals probably originated in Asia and moved into North America and Africa. The disappearance of the dinosaurs marks the end of the Mesozoic era and the Cretaceous period, approximately 65 million years ago. The primitive mammals continued to evolve as the continents drifted apart during the Mesozoic era. However, they remained small in size and number until the extinction of the dinosaurs offered the mammals new ecological opportunities. The fossil record shows an adaptive radiation of placental mammals in North America, Eurasia, and Africa. A similar radiation of marsupials also occurred in South America and Australia. This adaptive radiation of mammals continued through the Tertiary period and the Quaternary period of the Cenozoic era. During this era, which spans the last 65 million years, India merged into Eurasia, and the continents reached their present positions. South America remained isolated for most of this time period. Between 3 and 3.5 million years ago, during the Pliocene epoch, the Isthmus of Panama rose above sea level, connecting North and South America. Placental mammals moved into South America and largely displaced the native marsupial mammals. Only a few marsupials survived. Among them, the tough, opportunistic opossum moved out of South America and colonized North America. Australia, on the other hand, has remained separated from any other continent for the last 50 million years, and its marsupials remain abundant and diverse. The Earth's plates, some of which include continents, continue to move slowly. Presently, however, the distribution of species is being altered more by the action of humans.
Animations by Illustrious Incorporated, San Rafael, California.